Good afternoon. I'm Michael Wyman, CEO of Beeline Therapeutics. One of the most exciting new fields of medicine today is harnessing the power of our own immune systems to attack disease. Our immune systems can turn on, responding to infections. They can also turn off, protecting our own healthy cells. A lot of cell therapies today focus on turning on the immune system, deploying killer and helper T cells against cancer. But what about turning off the immune system? That's largely been overlooked by the field of cell therapy so far. And it's the turning off that holds the key to attacking disease caused by an immune system that's more active than we'd like. That's the focus of T regulatory cells that regulate other immune cells in our bodies. For example, a killer T cell can mistakenly recognize our own healthy tissue as dangerous, attack it, cause cell death, leading to disease symptoms like inflammation. A Treg may be ineffective at suppressing the response in this case. At Beeline, we're supercharging the Treg, empowering it to find and bind the healthy tissue. And once it gets there, to release cytokines that interfere with the killer T cell's activities. We're taking T regulatory cells, Tregs, out of the patient and then modifying them to better localize and to function better, and then putting the modified cells back into the patient. And specifically, we're making two key changes to the cell's DNA. First, we're putting in a targeting receptor, a chimeric antigen receptor that lets the Tregs localize to the right place in the body. Second, we're dramatically increasing the Treg's ability to secrete, to make cytokines that help the Tregs suppress the immune response. Scientists have been trying for decades to put these same cytokines into patients by mouth and by injection. The results typically haven't been very good, largely because not enough of the cytokine ends up locally, near the site where the immune system's too active. And if you just keep putting more and more in, there's too much toxicity, broad immunosuppression. Putting in a localized Treg lets us transcend this problem. The Treg can localize to the right place, and then it can release the cytokine there. We're setting the stage for a living drug that can sense its environment and know how much of the cytokine to release. We're cutting the DNA with an enzyme and knocking into a genetic locus without interrupting the gene's normal function. Only the cells that are fully edited successfully go back into patients. We're setting up a cell therapy that is safe, predictable, and tunable. Unlike random viral vector integration, we choose precisely the site in the DNA we want to knock into. Expression of the therapeutic we knock in will adopt expression of the gene we've chosen to knock into. And from the thousands of genes expressed in Treg, we choose the one with just the right level of expression and regulation to tune therapeutic expression, borrowing on the natural regulation of our genes. And we're one of the few groups anywhere in the world that can operationalize this approach with training from some of the world's leading labs at non-viral T-cell engineering. So that's the concept. Let's talk about what we've done. We've successfully knocked in a chimeric antigen receptor. You see in this flow cytometry data a population of cells expressing a CAR. We've created a cell that can localize, and that's the first step in our program to develop novel targeting receptors that will separate us from competition. And we've successfully knocked in an immunosuppressive cytokine. Here in this flow cytometry data, you see the population of cells expressing the key immunosuppressive cytokine IL-10. We've not only expressed it, but secreted it, which is what you see in the histogram on the right. And to the best of our knowledge, we are the first therapeutics company to publicly announce that we've knocked in, overexpressed, and secreted this key therapeutic protein out of a Treg. So that's the technology that we'll combine to fuel our platform. Let's talk about how we'll apply it. There are a host of indications we can address. Transplantation is the perfect first target. Here, our immune system can cause us to reject the transplanted organ that's been put into us, and the role of Tregs in inducing tolerance is well characterized. And transplantation is a massive market that can transform patients' lives. There are more than 21,000 kidney transplants in the United States every year. That number could actually double with better solutions. Today, we use non-specific drugs that suppress the patient's immune system to prevent rejection. But those same drugs suscept the patient to infections and even cancer. One of the two leading drugs is designated a Group 1 carcinogen by the World Health Organization. And those drugs don't even work well for all classes of transplants. Using a Treg to induce tolerance can unlock transplanting other organ types, like pancreatic islet transplants, where more than 1 million potential patients today can't get the transplants due to the risk that the islets really won't survive in the patient. In our short time here at IndieBio, we've achieved our key technical milestones, and in the next 18 months, we've mapped out developing novel targeting receptors and a series of in vivo efficacy and safety studies to get us to the clinic, where we'll move fast, leveraging FDA breakthrough and fast-track programs. Our proprietary technology is protected by provisional patents on compositions of matter methods filed with Wilson Sonsini. And we've assembled a superstar team to get this to the clinic that includes Dmitry Simeonov, who trained at one of the world's leading labs for non-viral T-cell engineering, Chris Chavez, who's a CAR T-cell engineer from Adicet Bio with training at Stanford, and myself with over a decade of business development experience. And we've attracted a scientific advisory board that includes two of the world's leading immunologists, a cell therapy industry veteran, the chief of pediatric transplantation at UT Southwestern, and the man who pioneered islet transplantation and remains the leader in the field today from University of Alberta. Cell therapy is currently the future for treating autoimmune disease and transplantation. Help us make it more of a reality today. Thank you.